Our next talk is by James Overton, and he will tell us about history and philosophy of the Immune Epitope Database. James, it's all yours. Please go ahead.
All right. Yeah, great to be here. All these wonderful talks today. Very excited about it. I'm going to talk about a project that I've been involved in for about a decade, and yeah, try and draw some connections to the other talks today. So this, when I saw the call for papers, I was quite excited about this. Let's see, I'm going to move this up. Quite excited about this, bringing together all these different communities to talk about, big data approaches in history and philosophy of science. That seemed just super cool to me. And so the contribution that I thought I could make is based on like my firsthand experience with one contemporary scientific database in particular, although actually I've contributed to several of them. And the kinds of things I'm hoping to show you are how scientific databases are like built and maintained and improved, how scientific ontologies are used to structure data in those databases. And then at the end, I want to suggest that these databases and the ontologies are interesting subjects like of themselves for HPS work. And the keynote talk this morning was really amazing. That kind of historical and more social database is more relevant to the projects that we've been talking about today. But yeah, my experiences with several different scientific databases, and so I thought I would make those kinds of connections. And so I'm going to start just talking about me. I'll talk about the Immune Epitope Database. I'll spend a bunch of time on the Open Biological and Biomedical Ontologies, which is a project I've been involved in for about 15 years now. Just draw some quick lessons for HPS stuff, and I'd like to finish early, so well, we can ask and hopefully a bunch of questions because I'm not sure exactly what aspects of my work will be kind of most interesting for this audience here today. Yeah, and I do have a PhD in philosophy, but I'm not really making any philosophical arguments today. I want to show you these things, and then I'll make some suggestions. I've been working as a consultant for scientific databases since I graduated my PhD in 2012. I work on open source and open source software and open science, and I make a living doing scientific data integration and mainly using these ontologies to do that. I have a little company called Notion Inc. Based in Toronto, which is another reason I was excited to make all your acquaintance here. And it's mostly just me, but I've had as many as 5 different subcontractors working for a dozen clients over the past decade. And IEDB, which I'm talking about the most, has been my biggest client over that decade. Yeah, I did a PhD in philosophy at University of Western Ontario under Bob Batterman. My thesis was on explanation in science, where I took a year of the journal Science and I did a bunch of text mining and empirical kind of approaches to that, including like randomly sampled tiny case studies. And I got a lot of skepticism at that time. I mean, the text mining stuff was published, but I got a lot of skepticism at the time about any kind of empirical methods in philosophy. And so I'm really delighted to see all sorts of great kind of empirical and empirically informed work here today. So that's been wonderful. And then the last little bit of my background is that I have been writing software since about 2000 in summers and part-time jobs when I wasn't in school. And on the other hand, I'm going to be talking about immunology a bit, but I'm not a scientist. I'm not an immunologist. I'm not a historian. So I'll try to answer your questions if you ask me, but those aren't my bits of expertise. These days, I used to call myself like a computer guy. Now I'm a bit more of a project manager. I used to write software. Now I write emails, but that's how it goes. And just so that you know, yeah, I get paid to work for La Jolla and on IEDB and OBO, all the things that I'm going to be talking about. I don't think that's a conflict of interest here, but so you know. And yeah, I contribute to other scientific databases and other ontologies for other clients. But of course, these views and opinions are my own. So Immune Epitope Database. Well, an epitope is just whatever is recognized by the adaptive immune system, which is present in vertebrates and allows us to learn immune responses in the course of our lifetimes. And most epitopes are just fragments of proteins from a bacterium or a virus that's invaded the body or infected a cell. And that full protein is considered the antigen. And epitopes are interesting because they're good targets. They help you make targets for vaccines. They help manage transplantation. They can be used in tags and proteomics and a lot of different things. And the Immune Epitope Database and Analysis Resource, or just IEDB from now on, is a database that's funded by NIAID, which is one of the National Institutes of Health in the United States. And it has a mandate to curate all publications on allergy and immunology, including things like autoimmunity and transplantation, but excluding cancer because that's under the purview of the NCI. And then recently, LJI got funding based on IEDB to start curating cancer things, so that the mandate was expanded by additional funding. And they have curated almost 24,000 publications, which is covering more than 95% of all publications in immunology from 1960s, the origin of the field, to the present day. That includes 6.5 million individual assay measurements and their assay conditions that have been curated into the database, and 1.6 million logical epitopes in the sense of like structurally distinct epitopes. So it's a lot of data. On the other hand, it's just a few gigabytes, the kind of thing that you can fit on your phone. So is it big data? I'm not sure. It's definitely big in terms of the investment that's been put into this. And all that data is freely available to browse and download. And I'm going to show you the website in a minute. So IEDB was set up in 2003, 2004, based on funding from NIAID. That funding has twice been renewed out until 2027, I think, at this point. And that's a competitive granting and renewal process. The co-PIs on the project are Alessandra Setti, who's a kind of legendary immunologist, and Bjorn Peters, who I work with almost every day. Bjorn runs like wet labs and dry labs, and in the sense of actually doing bench science and also doing a lot of computational stuff, predictions and analysis. The IEDB is hosted by the La Jolla Institute, and it has been since its inception, but in theory, a renewed grant could send the database somewhere else. And La Jolla is a private nonprofit that's located on the UC San Diego campus. It has many other different immunology projects. It's a big place with lots of money going through it. And Bjorn and I work on a few other different immunology related database projects through La Jolla. Just to put this in context, the kind of wider recent history is that there was an explosion of scientific databases in the kind of 90s and early 2000s as just the promise of doing this kind of doing this kind of work exploded, and the technology got kind of cheaper and more accessible. But and the kind of a Cambrian explosion, the funding ran out on a lot of these databases. And so some of them disappeared and some of them were incorporated into the survivors. And that's got to be an interesting history all by itself, because a lot of these databases come with very different models on what was going on, and their scope and their scale are quite different. And so merging two databases together kind of after the fact is kind of a fascinating little microcosm. But also just the scale of, we talk about big data in science, the scale of data being generated, and then the scope of these databases has continued to grow. They're increasingly important. Funding agencies mandate database submission in many cases. For example, the NIH has a policy that just kicked in earlier this year that every grant needs to come with a data management and sharing policy in the application. And that's going to explain how your data is going to be coded, managed, submitted to some kind of long-term storage archive, and shared with the wider community. And that's developing out of the FAIR principles for open data sharing. In particular, when NIAID funds research, that research has to go into IEDB. And then there's another path where IEDB curators are curating new publications from the literature. And just in that wider context, many scientific studies really do start with database searches these days, in some database like the IEDB, just to figure out like what is out there and what has been studied and where the gaps might be, what the promising candidates are for whatever hypothesis generation. And then they do the science, and then the data ends up going back into a database. And so in many ways, scientific databases are the beginning and the end of individual studies these days. IEDB employs a curation team of specialist curators. They have PhD level education in immunology. There were early attempts to try and get multiple undergraduate level trained immunologists to do the curation and get kind of cross-curator agreement that would be kind of good enough and I don't know, coordinate. But those failed. And so we end up with, I think about a half dozen curators are currently employed. They follow an extensive set of guidelines that they developed, and that's all publicly available, which is kind of amazing. So here's like it's like a Wikimedia page with the curation manual. You get a sense of it. It's probably 100 or more pages. When you actually try and print it out, there's a history here. When I looked at it, it went back to 2008. So it's kind of like a fascinating document talking about like how you bridge a scientific publication into the conceptual practical model that is the Immune Epitope Database, and all the edge cases that they've run into and the rules they've come up with for navigating those edge cases and getting kind of consistent curation to turn, you know, a whole orchard full of apples into into a database of consistent data that you can query in a consistent way. There's a limited use of machine learning tools for kind of abstract selection and classification from the literature, just to see what's relevant and what's not. But the actual curation is done manually. And so people like me work on improving the curation tools to help the curators work more efficiently, because they are expensive, of course. And so they have a custom web application that they use to enter their data. And then they also have different spreadsheet-based methods for putting in larger sets of data. And the data that comes in just gets larger and larger all the time. It used to be a paper might contain a handful of epitopes, and these days it's easily hundreds and thousands or tens of thousands, even hundreds of thousands for certain direct data submissions. But I would say that like medium-term prospects for fully automatic curation are pretty low. And if you look at the complexity of that curation guide, that would start to explain it. So I'm going to give you a quick demonstration guided tour of IEDB. You can do all this stuff at home if you want, just go to iedb.org. But we start on this search interface, which gives you a bunch of different axes that you can start searching on. The epitope itself, if you know what it is, different assays that are done on it, the source that that epitope came from, either the organism or the specific like protein that it came from. I won't explain MHC, the host organism that it was done in, which could be distinct from the source. If it's an infectious agent, then it's from one species into another species, or there's lots of transgenic stuff. And then the disease or maybe vaccination status that was done and that's the condition. So you hit search and you get to search results like these, which give you, 1.5 million records of different epitopes. And this one, for example, is going to be our writing example, synfecal. So this is the protein sequence acts as the name, and it's a particular, eight amino acids from this particular protein, which comes from a chicken. And it's been studied. This one's useful in different, I don't know in detail, but it's useful in different experimental methods and for kind of cross-checking data. And so it's been studied quite extensively in 221 different references and 563 different assays. And you can go online and you can buy this particular peptide if you want. And there's a bit more of an explanation of why it's useful. We can get a visualization in 3D of this. I think this is just a symmetric protein, and these two green bits are the synfecal epitope. And so again, it's just like a section of this protein that would be recognized or locked onto by different aspects of the immune system, either by antibodies or maybe chewed up inside a cell and then presented on the surface of a cell and then recognized by a T cell. And we can also go to the antigen tab. Here I've filtered on just chickens. So this is all the different proteins in a chicken that we could care about. And we're going to look specifically at GALD2. And we've moved over to this immunome browser view. And so this is that particular protein stretched out by the counts of the amino acid chains. And when we looked at this synfecal before, it describes it as positions 257 to 264. And so we see here, that's this this little section here, this bump, and that's just showing that a whole bunch of experiments have been done on this particular segment of this protein, and representing epitope assays of different sorts. And this is showing that a whole bunch of those were positive, and then there's been a whole bunch of different experiments that were negative on different parts of that protein. And yeah, and then I want to show you a few other things. If we go back to the search interface, we can pick on these finder buttons, and this is stuff that I work on directly. So for example, we jump to this finder, and this is like all the organisms that we have data on. So we can pick on like chickens. We can find all the text matches for those chickens. And then we're opening up this tree view, which is derived from the NCBI taxonomy, which is an ontology for taxa. And so this kind of tree view allows us to drive hierarchical searches. So we can do searches for all epitopes in birds, all epitopes in like frog or toad or whatever, mammals, all these different facets that we can drive our search by. And then I'm responsible for building these trees by combining and recombining and pruning different ontologies. All right, and we can skip over to the assay view. And then I just want to show you, when you select a particular assay, we get all this detailed information about it. So this is the information on the reference itself, details about this particular epitope. And then these are like textual definition or labels, but these are links out to different ontologies and other databases. So GenPept records of the protein, the particular taxon for chickens, this is a cross-genic transgenic mouse that this was experimented in. And then we have the experimental conditions under which the epitope was discovered or tested. And in particular, we have like tritiated thymidine. So we jump over to, this is a term from the Ontology for Biomedical Investigations, which is an ontology that I work on a lot. And so we have a tree of different assays, and these are immune epitope assays, and then T cell ones specifically, getting more specific. And then this one in particular is measuring 3H-thymidine, proliferation of T cells. And so here, it's a bit small. I'll make it bigger. We have a logical definition of this particular term using a language called the Web Ontology language. And then we also see like cross-references for other ontologies that are using this. And we have a few different ways to view it that I'm going to skip. But OB itself contains about 5,000 terms. This is a different view of the same thing. Contains about 5,000 terms. We focus on like assays in particular. There's about 1,600 different assays, and we logically define them and give them textual definitions for humans and synonyms and all sorts of different things that you can use to drive different kinds of searches and browsing and analysis. And OB is just one example of the OBO Foundry ontologies. So I'm going to skip there now. So OBO is a community of open ontology projects with a set of shared tools and infrastructure that I've contributed to quite a bit over the last couple decades. OBO includes more than 200 different active projects across a huge range of biology and biomedicine. And I'll just scroll for kind of shock value. And our best principles and best practices are all about kind of interoperability between those ontologies, so that you can have your data that links between them, moves between them. They can be cross-defined using different terms from different ontologies in the same logical axioms. And then the upshot is you get back to these kinds of records for a particular assay that was done that's linking in off in all these different directions. And ultimately, you could take this kind of record and you can integrate it into other databases that are using the same ontologies, the same controlled vocabulary systems to analyze their data. So let's see. That was the demonstration I wanted to give. just to be clear, these scientific ontologies are not necessarily what philosophers mean by ontologies, but it is what Grace is talking about earlier. So kind of like terminology systems for organizing our knowledge, developing out of the knowledge representation branch of artificial intelligence research. Yeah, and they combine human-readable annotations with machine-readable axioms. And my job is to build computational artifacts out of those things, which we do using open platforms like GitHub. And I'll talk about that in a second. I'll skip the technology stack. I talked briefly about OBO. The core of OBO is the Gene Ontology. I would say it's kind of, we grew out of that project, and the Gene Ontology has had like unbelievable impact on all sorts of biological science since the 90s. But just to be clear, our ontologies, like we have millions of classes. So the terms often get very specific, like 3H tritiated thymidine cell proliferation assay of T cells. They're all kind of common nouns for science, but each one has like a globally unique identifier that we can use to distinguish them. And it covers a whole bunch of different domains, which is why these the interoperability is so difficult, but so valuable to connect things like anatomy up with I don't know, agriculture. And we have a set of kind of open source principles that drive our interoperability, but we can come back to that later if you want. And then the last few points I want to make is OBO and IEDB, we're doing this open science, open work, like largely online. So that means that a lot of this work is done in open source repositories where we have a full history of like issues, requests for changes that are actually made. And then we also have like email lists and trackers, version control logs, meeting minutes for all these things. And sometimes these go back 20 years. So this week is the 20th anniversary of the OBO Discuss mailing list. You can look at everything that anyone has said on that list in 20 years. And for like HPS people, this is like an embarrassment of riches. If you're interested in like recent history of science, kind of contemporary science, then I suggested you need to be taking advantage of some of these riches that are just out here for the taking. And yeah, and if you're interested in projects about the more distant past or the history of kind of intellectual history, then I think there's lessons to be learned about how these scientific databases have grown and evolved, and the mistakes they made and how they fix these, and also how they've used, in particular, these scientific ontologies and community terminology systems to drive quality assurance and interoperability. So yeah, we have a few minutes for questions, I hope. And these are questions you could ask, but we could follow up later. Yeah, thank you very much.
Thank you, James. Thank you very much. I'll go ahead and ask one that I had in mind. So I see that there's a lot of practical expertise here on your part and part of your colleagues. Of course, the database is in a field that's not intellectual history, of course, although the output of it, as you mentioned in your final comments, the final comments can be used as a source source for us historians to learn about the belief system of this community, at least in the last 20 years, and also how it evolved. I can easily envisage us tracing changes in their ontology. If that's documented, I believe it probably is documented. So those things aside, when it comes to database building itself, which of the tools or functionalities or solutions that you or your colleagues have been implementing, which of those you think might be directly transferable to the field where the task is to document how epistemic agents take epistemic stances towards epistemic elements? Like, is there any, I understand it's a very broad question, but what would be some of the things that come to mind first?
Mm-hmm. Yeah, I've watched a couple of presentations on that your project that I found on YouTube and just discovered this week as I was preparing. Yeah, and coming up with the kind of the top level ontology makes a lot of sense. And Grace's presentation was a great presentation. The keynote this morning on experience with other kind of historical databases, that's exactly on the right track. And some of the things aren't relevant because none of the databases I work on are temporal. We curate historical like scientific records, but we don't yeah, we don't care about well, I don't know. It's just not the same thing. You can go back and look at references from the 60s, but we treat them the same as we treat them, as we treat contemporary records. So, but yeah, I think you're on the right track with all these things, coming up with the right schema. And I guess maybe the one thing I've noticed is a bit too much hesitation. That you kind of just have to try these things and see if they work and see if they don't. So for example, the IEDB has gone through 3 different schemas in the history of the database. And so it had an original schema, which was insufficient. And then there was another one, IEDB 2.0, and then a smaller revision for IEDB 3.0. They published all these things. So if you wanted, you can go on the web and see those 3 different editions and the changes to the schema. But there is just a matter of evolution in these things, and you're not going to get everything right the first time round. And we run into questions of scope a lot in our projects. When we start top down, we often make mistakes, and we are just too general too quickly, and things get too philosophical. And then when we start bottom up, sometimes we just kind of get too hesitant to make the big changes we need to do to restructure things. And so, I don't know, we can talk more about practical advice, but those are a few things I'd say off the top of my head. You do have to, you've done all the preparatory work, I think, and you need to dive in and start doing prototyping, and then just give yourself room to make mistakes and learn from those mistakes iteratively, because you're not going to get it right the first time.
Thank you, James. I'm looking forward to a continuous conversation on this topic. This is very illuminating. Christoph.
Yeah, thanks a lot for the presentation. I'm really happy to get acquainted also, James, because your paper, your 2007 paper, if I recall correctly, right on explanation, was one of the pioneering works on using this.
I think it was 2010, but it felt like,
or 2010. Okay.
Felt like 2000. Yeah, I don't know. It feels like a long time ago.
It feels like a long time. But anyway, it was real pioneering work in the in philosophy of science to investigate the usage of a specific term in different domains of science. So but it's also, I find it's a pity that you're no longer doing philosophy of science. But on the other hand, when I see the great stuff that you do, I'm sort of saying, well, it's maybe it's also for the better. And I was sort of curious, you say, okay, it's an embarrassment of riches for HPS people, but I was sort of wondering, which type of philosophy questions do you think could be asked fruitfully, by mining this database, either the IEDB or the OBO database. Of course, there are many very interesting questions as to how science pulls together big databases. And here I'm thinking that maybe this is the type of work like Sabina Leonelis has done, and is still doing. But I was sort of wondering, do you have like prototypical questions that would really be interesting for philosophers of science that the type of databases that you mentioned may be amenable to answering?
No, it is a really good question. I mean, Sabina Leonel has done amazing work. There's this kind of ethical side of things that I never feel well attached to figure out, but setting up these ontologies is forcing a view on the community that they are getting required to adopt. And these aren't like elected leaders or anything, they're kind of self-appointed leaders. And there's the consensus building process is kind of really interesting. So there's sociology, ethical questions. And then I think we have, the other questions that are interesting to me are kind of fine-grained theory change and acceptance elements. One thing is the ontologies tend to encode I would say they encode accepted knowledge more than like pursuit kind of knowledge. Most of the stuff I work on is quite conservative in that sense, because they don't want to go back and change things, so they don't get out ahead of the theory. Maybe unlike the high energy physics stuff we talked about earlier today. And so there's some differences there. There's probably some differences in just how different communities approach these aspects. So there's a wide range of communities using the same tools, and so there'd be kind of comparable artifacts across these things. But yep, I don't do this anymore, so it's up to you guys to come up with the good questions, but I will help lead you to some answers if you're interested.
Thank you very much, Christoph and James. I can envisage several fruitful ways in which this can be used, and you mentioned some of them. Thank you very much again.